Australia.
And this place nurtured my passion for science and nature. As you can see here, we have a picture on the right-hand side, which is a picture of a snake poo. This is gigantic snake poo, was located near our chook shed, and I was terrified of meeting the snake that laid that gigantic beast. I love nature and all the diversity that can be seen in the marine environments, right through to our terrestrial environments. I look up to trees, even the Joshua tree you see there on the right-hand side. I'm inspired by trees. Their massive woody stature is propelled by decades to centuries of height and radial growth. No other organism does this. Trees are a fantastic organism to study. They can't run away. Trees remain fixed in the same position for centuries, and they endure many generations of insect and pathogen attack, while the tree's defenses have to keep up. We make use of forest products every day, from cradle to coffin. I did my PhD on a eucalyptus species that was used for structural timber, and I was tasked with analysing a population of trees and identifying the trees that had good wood quality and also trees in the same population that had poor wood quality. Further, I was tasked with identifying the single letters in the DNA code that meant that one tree would grow up to be big and strong with very good wood properties, while another tree would be small and skinny and had poor wood properties. After completing my PhD, I was then recruited to UBC to come and help identify a way to help the massive insect attacks that were destroying Canadian forests. For me, it seemed very foreign that just a small insect could be, in fact, destroying entire forests. Coming to Canada, the environment was very different to that of my homeland. I wasn't used to snow, and this was my opportunity to embrace winter. I could be a kid again, and first thing I needed to do was scribe my name into the snow. It was a very good project to start. There was lots of exciting people, lots of talented people to rub shoulders with. I got to travel over the province and sample trees and torture trees and poke them in the name of science. The goal of my postdoc was very similar to that of my PhD. Here, I was tasked with identifying the single letters in the DNA code that meant one tree would be small and susceptible to insects, while the alternate letters at the same position in the DNA code meant that another tree would be big and strong and resistant to insects. The goal would be for a tree breeder to sample seedlings and identify that they were insect resistant at that early stage. For my second project, I got to work on a very well-known species known as red cedar, and I was again tasked with identifying markers in the DNA code that predicted that one tree would have clean wood, while the other tree had heartwood rot. But after a number of these projects, I was starting to get bored with the academic process. I was getting tired with the culture of the constant whip of academia saying you need to publish or perish. I wanted to start my own genomics company and create my own destiny, and that's where I got put in contact with E at UBC. The customer problem was that outside human and cattle genomics, DNA testing was not routine. It was far too expensive, and it was far too complicated to expect a customer to go and extract the DNA for themselves or interpret and read the DNA sequence for the information that they were looking for. So this is where BlastSeq Genomics came into play. We were to set up a revolutionary lab in Vancouver that would put genomics in the hands of everyday people. It would put genomics in the hands of farmers and people that were regulating agribusinesses. The company would offer two different types of DNA testing services. The first was a low-density DNA fingerprint that could be used to resolve the identity of samples in a nursery that might have been mixed up. It could be used to determine the ancestry of a population of trees. The second service that we would be offering would be a high-density DNA profile. and this could be used for more complicated interpretations. However, I was a little bit concerned about working with agribusiness. What if there was a drought? What if the government changed their spending cycles? And living in Canada, will my samples actually be affected by the seasons? Canada is a cold place after all. So I needed to think of something that a genomics test that could touch more people. I was wondering, is online dating an opportunity? Can I set up a DNA-based matchmaking service? I was really excited about the science of pheromones, and there were recent studies indicating that if two people enjoyed the smell of one another, then they in fact had an opposite immune system. Well, if people actually despise the smell of one another's shirts, they're actually more similar than they thought. Could this be the basis of my new DNA testing service? It seemed very simple to take some spit samples from some single people and identify those that were not compatible, having the same immune system, and people that were perfectly compatible by having opposite immune systems. So I'll end it there. But if you're looking for any romance, if you are single, please check out dnaromance.com. And if you're in the market for some DNA testing, feel free to contact me at Blastic Genomics. Thank you.